hours, things basically change, and we go from one state of specification to the next one and to the next one. And one thing that rose my curiosity is really trying to understand the molecular mechanisms that underlie these dynamic processes. So I'm going to try to give you a few examples of that. So for us, not for the nervous system, but for us, things, I'll go with my arrow because the angle is ובאמת לא, and I'll dim the lights. I think this is what I should do. Is it better? Okay, so for us, things begin with the process of neurulation. This is a scanning electron micrograph of an avian embryo, in which you can see the formation of what is going to become the closed neural tube. And this neural tube is going to be the primordium of both the central as well as the peripheral nervous systems. And what you see here are these marvelous cells that begin leaving, exiting the dorsal portion of the neural tube. Can you see them with their longitudinal axis pointing towards these little cubes, the mesoderm? And these are cells that are going to leave the neural tube and going to become almost the entire peripheral nervous system, cells which are called neural crest cells. And during development, if we take a cross-section through this tube, through this baguette that I showed you just now, what you see is that there is this sequence of events going from neural crest that generate most of the peripheral nervous system to melanocytes, which are also a very famous and clinically relevant derivative of the neural crest. And then this dorsal area, the dorsal midline area of the spinal cord, which is the axial level that we are concerned with, will transform into a very important structure called the roof plate, which is an organizing center for development of more ventral cell types in the spinal cord and also in the brain. And then the roof plate will transform into radial glia, which will transform into dorsal ependyma, which will generate like stem cells of the mature nervous system. So we are in a kind of sequence of events that is highly dynamic, and we would like to understand the whereabouts of that. So let's begin with the neural crest. The neural crest cells are exemplified by a very dramatic process of EMT, epithelial mesenchymal transition. They are born as, soldiers. This is now fashionable. Soldiers sitting one adjacent to the other, bound by gap junctions, tight junctions, and all of a sudden, these cells, by a mechanism that we discovered many years ago, are able to dissociate from each other and undergo an epithelial to mesenchymal transition, a process that is very analogous to metastasis during tumor progression. And what we see here is a movie by Irit Choval, in which we see how these cells are able to basically detach from the central nervous system primordium, adopt a mesenchymal phenotype, like a fibroblast type of phenotype, and go all over. Obviously, the routes and the pathways of migration through which these cells go are very, very stereotypic, equal from embryo to embryo, a little bit different at different axial levels, and characteristic of different species. All of them have been already fully characterized. But then, when we move to older stages, no matter what is the gene that I show you here, this is an in situ hybridization, an RNA-RNA reaction. The contrast is not very well in this barcode, so I don't think you see everything, but you go from the neural crest stage and just look at the dorsal portion, which is marked here. Okay, this is the area of the tube we are going to deal with. And then you go to the roof plate, this dorsal midline structure, and then the roof plate will elongate and turn into what we call the spaghetti-like structure, which is called the radial glia. And all this process is concomitant with the shortening the stretching of the lumen of the tube, which is very wide, to form the final central canal. So there is atresia. There are a lot of processes that we are studying now regarding these particular events, but this is just an introductory slide to show you how we proceed, and this is the area that we are going to deal with. So when I talk about phase transitions, basically, I am going to address 2 out of these 4 main points. The first is a very long-lasting issue in developmental biology of the nervous system. How do neural crest subsets segregate from each other? How a multipotent cell will become a neuron or a glia? How will it decide to become a glia or a melanocyte cell? And we have amazing data, this will be for the next time, regarding the segregation between glial and melanocytic phenotypes. How do neural crest cells end their journey? For many, many years, my lab has worked on the onset of migration, the pathways, the mechanisms of migration of cells. But now, at the almost end of my career, I decided to shut up, to finish crest emigration, and try to understand what happens later on, a subject which is quite a niche, not addressed by other laboratories for the time being. So how do crest cells end their journey, and how the dorsal portion of the tube, this triangle of the midline, transits into the next phase, the roof plate. Basically, this concerns what I wrote here, the separation between the peripheral and the central nervous systems, whose origin is the dorsal neural tube. Namely, both branches of the nervous system originate in the same place, but they have to separate at a certain point. Third, how do roof plate cells separate from dorsal interneurons? We have roof plate cells. In a minute, you will see what I mean. And immediately ventral to the roof plate, you have 6 different types of dorsal interneurons that generate information in a commissural manner and go to the brain, but also connect to the dorsal root ganglia, and so on and so forth. So this is question number 2. There is a boundary between the roof plate and dorsal interneurons. And finally, how do roof plate cells transform into radial glia, ependyma, and so on and so forth? So I am going to address points 2 and 3 today. Different people in the lab work on these different topics, and let's concentrate on how crest cells end their journey and how do we transit into the roof plate. So what is the crest? It generates most of the PNS. These are highly proliferative cells, undergo EMT, and they laminate and migrate. But all of a sudden they stop, and we are left with a roof plate that constitutes the definitive dorsal spinal cord, namely the CNS. They act as an inducer via molecules that we know already of dorsal interneurons, regain pseudostratified epithelial properties, do not delaminate, and they exit the cell cycle. So look at the yellow cells and look at the blue cells. The yellow cells are neural crest cells, and when these cells delaminate sequentially, what we see is that the roof plate cells progressively go from a ventral to a dorsal position. Namely, they relocate ventrodorsally to adopt their final dorsal midline position. This work is 5 years of work of a PhD student who did single cell analysis, fate mapping in time and in space, because time and space are interchangeable values at these points. So basically, we know that the roof plate is originating ventral to the cohort of pre-migratory cells, and once these cells emigrate from the tube, roof plate progenitors undergo this ventral to dorsal relocation, and they end up in the dorsal midline of the neural primordium. So when this happens, what are the biological correlates? If we look at the roof plate, we know that the roof plate throughout the axis is quite different. So for example, if we look at the hindbrain, the area that is going to generate your cerebellum, medulla, we see that the roof plate is extremely wide in contact with the rhombic lip, And the lateral part of the roof plate will generate, for example, the choroid plexus, okay, which is responsible for synthesis of the CSF. At the trunk level, which we deal with, the roof plate will generate radial glia, dorsal ependyma lining the dorsal portion of the central canal, and so on and so forth. And at the tail region, the lumbosacral region of the axis, what we know is that only in avian embryos, chick, quails, birds, everything, there is a very, very specific structure generated from the roof plate, which is called the glycogen body. It's like a kind of tumor that arises from the roof plate. Cells proliferate, have a lot of glycogen, and we believe they behave as a kind of barrier for axonal decussation, for contralateral axonal elongation. So the roof plate is important. It is without a roof plate, as you will see later on, there are no dorsal interneurons, and they are obviously a kind of midline, pretty much like the floor plate. So imagine that you're building a house, okay? You need, first of all, to build the building blocks. They will determine the basic pattern onto which we are going later on to put the toilets and to put the kitchen and the windows. This is a kind of scaffold like that, the roof plate. So now what do we understand about formation of the roof plate, end of crest migration, to try and understand what causes the end of migration of crest cells? Let's start by understanding what causes the beginning of migration. And I will summarize it very, very quickly because, as you see in the references, this is quite an old work from the lab. We found that there is a molecule called bone morphogenetic protein, BMP, very important for the rest of the talk, initially found as a bone a factor inducing bone growth, but now we know that it is essential for the specification of the neuroectoderm. And we see that BMP is homogeneously expressed throughout the entire dorsal neural tube. But there is its friend, an inhibitor of BMP called noggin, which is expressed in a graded fashion, very concentrated in the tail, and then less and less until it disappears later on. And this creates a gradient, a counter gradient of BMP activity, by which BMP activity is very low here, but begins to be very high in these areas. And this coincides with the onset of emigration of crest cells. So by doing functional experiments in chicken and in mouse, we could demonstrate that there is that BMP is a master regulator inducing delamination of crest cells, that there is a counter gradient, a kind of crosstalk between BMP and its own inhibitor that creates this gradient along the axis, which tells the cells when to begin migrating, and I'll leave it there because it's quite a complicated story. So for us to understand, BMP is the master regulator, and then we uncovered a whole molecular network that regulates this process with downstream canonical Wnt activity, synchronization of the cells to the S phase of the cell cycle towards delamination, and so on and so forth. So for us now to remember is that BMP is essential for neural crest DMT. And if we want to look at BMP activity, one way to look at it is by phosphosmad 158. This 9 is an error. BMP phosphorylates in the cytoplasm a molecule, an intermediate a transitional protein called SMAD. And we have antibodies against the phosphorylated form of SMAD, which says if it's positive, it means that there is BMP activity. And what you see is that at the level of the crest, this is roof plate, BMP activity occupies the entire pre-migratory domain. But BMP loses activity, or the cells lose responsiveness to BMP at the roof plate stage. The roof plate does not respond any longer to BMP. What continues responding? DI1 interneurons. You have DI1, 2, 3, 4, up to 6. DI1 interneurons are known for a long time to depend on BMP and Wnt signaling, and we see that this is the case. So having looked at that, what we see is that indeed crest markers, one of them is FOXP3, a transcription factor, is expressed in the pre-migratory crest, but it is lost in the roof plate. So there are markers that are characteristic of crest cells, others that are characteristic of roof plate cells. One of them is R-spondin. R-spondin, or roof plate specific spondin, is absent in the crest stage. It begins to be expressed in the roof plate. And thanks to an initial bulk transcriptome done by Chaya Kalcheim in my lab, we could find like dozens of molecules which are specific either to the neural crest, pre-migratory stage, or to the roof plate, which nobody knew before, and gave us now a tool, a kind of readout to be able to study end of migration and differentiation of roof plates. And another molecule that is going to be key to this talk is RALBH2, okay, which is not expressed in the crest. It begins to be expressed in the roof plate. And also RDH10, CRABP2, no matter the names, these are all enzymes or transporters belonging to the retinoic acid pathway, vitamin A pathway, which at this stage begins to be synthesized in the dorsal neural tube. Namely, all of a sudden, when we transit to the roof plate stage, the dorsal tube begins to synthesize retinoic acid. And basically, what we see is that there is a kind of replacement of BMP signaling that stops being active there, and RA begins to be active. So, because of many reasons I don't have the time to elaborate on, we predicted that retinoic acid may be important for this transition via inhibition of BMP signaling. Okay, so bear with me. I'm not giving you the reasons because it would be too long. So in order to analyze such a hypothesis, what we did is we adopted a loss of function approach by which we inhibited locally only in the dorsal neural primordium retinoic acid signaling. You can do it by different ways, all of them very well proven. We did it by two means, by misexpressing in the embryo. We do it by electroporation in the egg, in ovo. Most of this work has been done in quail embryos, which are very small and very easily amenable to manipulation under a binocular. So we misexpressed CYP, which is the enzyme that degrades retinoic acid. Alternatively, we misexpressed a dominant negative construct, a DNA encoding a dominant negative construct of receptors of retinoic acid, which are quite a story. And under these conditions, we can, in a cell-autonomous manner, inhibit retinoic acid activity. You see the controls here. This is GFP. We always put a GFP or RFP control to know that this part of the tube has been indeed electroporated, okay? And you see the activity of a retinoic acid responsive element, of a reporter of retinoic acid activity. This is under controlled conditions, but in the presence of either of the inhibitors, retinoic acid activity is gone. You can see it here with alkaline phosphatase as well. So we have two beautiful tools to inhibit locally retinoic acid activity. And we do it, as I said, by electroporation. You open a window, for those of you who never saw it, you open a window at day 2 in the egg, and you can put this poor embryo into a kind of electrical chair. You, in between two electrodes, DNA is negative, you inject it into the lumen of the neural tube, you zap the embryo, DNA will go from the negative to the positive pole, and it will create this electroporation, as the word says. It will create tiny microsecond holes, pores in the cells, through which the DNA will enter, and then will be obviously transcribed, translated, and so on. So we can do a beautiful gain and loss of function studies using this technique, and the first result is the following. So you remember these two images. You remember that at the crest stage, there is, this again are the controls that the area has been electroporated, okay? So look only at the upper panels. In the neural crest, activity of BMP, positive. In the roof plate itself is negative. But if we inhibit either with dominant negative retinoic acid receptor or with CYP, we extend the period of BMP activity, namely the cells continue to be responsive to BMP. Basically, what we do is we can say that retinoic acid activity is necessary, okay, because it's a loss of function experiment, necessary for inhibiting, for the timing of inhibition of BMP activity in the dorsal neural tube. Now, question number 2, how does retinoic acid inhibit BMP activity? Now, you remember that for crest cells to go out from the tube, we had a kind of counter gradient between BMP and one of its inhibitors. Now, the story comes back again. This is why I told you also that part. We found in this transcriptome by Chaya that there are certain molecules which are inhibitors of BMP, like BAMBI, okay, and herin. Now, BAMBI is not expressed in the crest, it is expressed in the roof plate. Herin is also expressed in the roof plate. None of them is expressed at the neural crest stage. So one possibility is that retinoic acid, okay, is able to upregulate, this is a hypothesis, to upregulate HERI1 or HES, it's the same, BAMBI, gremlin, and additional inhibitors that we find to be expressed very specifically in the roof plate. So let's test whether this is indeed the case, and it is. So what we see is that if we inhibit either by CYP or by dominant negative, BAMBI, which is expressed in the control, fails to be upregulated under those conditions. So retinoic acid is necessary for the onset of expression of inhibitors of BMP. Thereby, maybe this is one way in which retinoic acid stops or brings BMP activity to an end. You can see, by the way, all the quantifications always present in the very same slide. So is this true? If we upregulate a certain inhibitor of BMP, are we going to inhibit BMP activity in the process of emigration? This is true if at the stage of crest cells we misexpress, we overexpress either BAMBI or gremlin or HES, each of these which are expressed normally only in the roof plate, but if we advance, if we do an early misexpression, we see this is the control, this is phosphosmab, and you see that phosphosmab is reduced in the hemitube that has been electroporated. Moreover, crest migration has stopped. You see all the cells do not die. You see them concentrated in the tube, but these are DRGs, these are Schwann cells, and they lack the contribution of the cells that have been electroporated. The same is true for day 4, so no delay in emigration. The cells cannot emigrate when we apply an inhibitor of BMP. So what all this means is that in the absence of retinoic signaling, BMP inhibitors, which are normally expressed in the roof plate, fail to be upregulated. Consequently, BMP activity extends into the roof plate stage. Again, yes, Raunak.
So, I'm just wondering, how does this signaling exactly take place? Is retinoic acid a secreted molecule?
Oh, absolutely.
So that means that one cell is secreting it, the other has a receptor for it, which is then, so the BMP is getting downregulated in the receiving cell, right? The cell with the receptor for retinoic acid. Yes,
retinoic acid acts via a heterodimeric type of receptors, which are RXR and RAR. These are nuclear receptors, unlike BMP, which are membrane-bound receptors. So you have to assume that retinoic acid is secreted from cell 1. It acts on cell 2, which is immediately adjacent to cell 1. It enters the nucleus, it activates or does whatever it does, and then it acts on BMP signaling. So it has to act via the nucleus.
Right,
yes.
but then would your GFP, the assay that you're doing, which cell is getting the GFP?
Most of the cells in the surrounding. We are doing a kind of massive electroporation of the area. Now, having said that, massive electroporation is still mosaic, so you don't have more than 70% of cells electroporated. You always have a few cells which do not get your GFP or your plasmid or whatever you have electroporated. Okay? Baruch?
In Drosophila, we do a lot carotenoid deprivation. Anyone tried it in mammalian or in vertebrates?
Not to the best of my knowledge. Okay? Ocello? Okay. So, again, I'm telling you the logic of the story so that I hope you can follow it. Okay? So, the prediction is that if we inhibit retinoic and BMP continues to be active, this means, for us, BMP means neural crest. So we would expect that markers of the neural crest will prevail at the roof plate stage, and that maybe neural crest cells will continue delaminating from the neural tube. And this is indeed what happens. So what we can see is that molecules like FOXD3, which is absent in the roof plate, continue to be transcribed in the roof plate of the treated embryos, of those which lack local retinoic acid. The same goes for SNAIL2 and for SOX9 and for a variety of additional molecules. So we continue basically with the neural crest traits, maybe at the expense of roof plate traits. And what is more important, cells continue to delaminate. So this is a kind of hemineural tube trick in which we see that only in the hemineural tube that has been electroporated with the dominant negative, both of them have been electroporated with control GFP, cells continue to come out at a very, very late stage. And in one talk that Dina gave a few years ago, Eli asked, what is the fate of these cells? We know now what is the fate of these cells. These cells are basically retaining a glia melanocyte progenitor kind of trait, a double progenitor trait, which is absolutely interesting for us, but I'm not going to elaborate on that. So the idea that is that retinoic acid induces the end of neural crest emigration via inhibiting BMP signaling. Now, I would like to make a little parenthesis and say the following. If roof plate traits inhibit neural crest traits, put yourself on a scale of time, okay? Is it possible that neural crest traits affect roof plate traits? And to verify this, and you'll see in a minute what I mean, we extended the period of FOXD3, SNAIL, and SOX expression. Namely, we gave an extra dosage of crest markers, and indeed what we see is RALDH, a factor, a synthesizing factor for retinoic acid, is not expressed in the roof plate. We fail to upregulate it. And the same goes for a specific reporter of retinoic acid, of RALDH, which is a roof plate specific reporter. What does this mean? This means that not only retinoic acid via inhibitors of BMP inhibit BMP, inhibiting crest production, but if you are still at the crest stage and you express FOX, SNAIL, SOX, and whatever, you also inhibit the onset of retinoic acid signaling. Namely, there is a kind of feedback, cross-inhibitory, cross-repressive talk that takes place not in space, it takes place in time. Meaning that if I am a neural crest, I will actively repress any possibility of any gene of the next stage to become expressed. And once I am a roof plate cell, I will inhibit all of the neural crest possible genes. So these cross-repressive interactions have been shown during development of the nervous system, in particular ventral interneurons and motor neurons, but only in space. And this causes the refinement of the fields of expression of motor neurons, VI1, VI2, and so on and so forth. But the temporal regulation of cross-repression, this is the first time that this is shown. So retinoic is necessary for ending crest production. Is it also necessary for the formation of the roof plate? This is the second part I wanted to deal with. And for the formation of interneurons. So are these interdependent processes or independent processes? And the first hint came from a work we did with Quentin Schwartz, Shai Ofek, and Quentin Schwartz from Adelaide, Australia. This is mouse work. So what we did is, I go back a step. We asked whether retinoic acid is important for the formation of the roof plate. The answer is no. Okay, why? We see, for example, SLIT1, which is a chemorepellent in the dorsal tube. It is downregulated in the presence of retinoic compared to control. But additional molecules, so I brought you here a positive control, as if this affects the roof plate. But if we look at ARSCO1 or ARSCO3, it doesn't change. At NORI, which is NDP, both of them positive regulators of canonical Wnt signaling, they don't change. If we look at Draxin, an inhibitor of Wnt signaling, it doesn't change. So we have a roof plate in spite of not having retinoic acid. But this roof plate is not normal because it also contains crest cells, as I showed you before. So if we look at this very weird roof plate, what we see under control conditions are spawning in pink, no snail. Snail is crest, ARSCO is roof plate. No snail. And the overlay shows obviously that these are in different places. But look at the treated ones. We have ARSCO and snail co-localized at the single cell level. So we have cells that fail to segregate and that adopt a kind of conflicting identity, being at the same time a roof plate cell and a neural crest cell. This does never happen normally. Again, this is SOX9, okay, together with ARSCO1, okay, and another example even of cells delaminating from the tube, co-expressing ARSCO1 and neural crest markers. What happens to DI1 interneurons? So I'll introduce you to them. This is the roof plate, this area, a very tiny area. And ventral to the roof plate, we find in dorsal interneurons. Now these are DI1 interneurons because they express a certain marker, a certain molecule, which is called BARHL1. And as you can see under normal conditions, there is a boundary, okay, which separates roof plate from DI1 interneurons. What happens if we inhibit retinoic acid signaling? You do not respect as a DI1 interneuron the boundary, and you colonize, you extend, you expand into the roof plate itself, and all the quantifications are here. So this means that this roof plate is a weird type of structure and that retinoic acid is necessary to segregate the different fates. Now, this was the time at which we said, okay, we need now a resolution at the single cell level to see whether indeed, okay, bioinformatically, molecularly, and so on and so forth, all these markers are in the same cell, and to that end, Dina Reckler in the lab performed a single-cell RNA-seq analysis in order to avoid all kinds of ventral cell types. We electroporated MSX1 citrine, which, as you can see, occupies only the dorsal third of the neural tube, so we could enrich for dorsal cell types, and with or without dominant negative retinoic acid, and compared the transcriptomes of both conditions. Here you can see the embryos. They were electroporated at day 2, fixed at day 4, and subjected to the 10x genomic, 10x platform. And what you see here is what we call the famous octopus, one of our favorite animals. This is a UMAP, a two-dimensional representation, graphic representation of clusters. For those of you who are less knowledge about that. Each one of the colors represents a cluster, which represents a different set of cell types, which are grouped thanks to known markers. And we are going to concentrate on the roof plate, but we also have DRG progenitors, melanocytes, and as I'll show you, different cell types of the dorsal neural tube. We have 23 distinct cell populations, and what was very important for us, for the first time, we have now an extremely rich source of many, many molecules that characterize the entire developing nervous system, also under normal conditions. As I said, we'll focus on the roof plate, but first of all, let me just give you the whereabouts of this UMAP, of this famous octopus. So time is the first value here. What we see, these are different genes, okay? Different genes expressed in different regions of the octopus, so bear with me. The head of the octopus is composed of proliferating cells, okay? It is composed of cells which express CDKs, BUB1, and all kinds of proliferating markers. It is composed of the proliferating cells of the neuroepithelium. Then we go to the intermediate, to the onset of the arms, of the branches of the octopus, and we see notch signaling molecules like JAG, delta, and so on, which mark intermediate stages in the specification of these progenitors. Then we go to neuro G, neuro D, and others, which go a little bit further in the branches. These are DI1, DI2, DI3, DI4, up to DI6, okay? So all these are different dorsal interneuron populations, up to postmitotic neurons expressing KIF26B, calbindin, and so on. So basically, we have from the head of the octopus up to the tip of the branches a sequence of time, of developmental time. Now we also have a sequence of spatial time, and I'll go very briefly through it. For example, this is dorsal because these are DRG and peripheral neurons. This is the roof plate, and this is DI1, DI2, DI3, as I said. So basically, and these are the markers, I will not elaborate on them. So the octopus presents us with the opportunity to have also a spatial organization of different cell types of the dorsal spinal cord. So this is extremely valid, and as I said, this is going to be the source of a lot of work in the lab in the future, in particular this. This is the representation of all the different clusters, and these are the genes that are expressed in the roof plate compared to the other clusters. So you can see a lot of specific genes, some of them expressed in other clusters to different extents, some others which are absolutely specific to the roof plate. Okay, so the dot is smaller or bigger depending on the percentage of cells in the roof plate expressing this particular gene. So without entering into any detail, the message is that we got a lot of novel markers that Nitsa in the lab is beginning to test by CRISPR-Cas loss of function in terms of their function in roof plate development. But let's go, I'll skip this, and let's go to our point. We can compare differentially genes that are upregulated or downregulated under experimental conditions. We have many, many, many of these figures. I'll just show the most relevant one. In yellow, you have the upregulated genes, so you have many genes upregulated under treated conditions, and another set of genes in the roof plate which are downregulated. So we get roof plate genes, okay, that are upregulated, and you can see, for example, how R-spondin 3 is upregulated in the bioinformatic analysis and in the validation by in situ. You can see DI1 interneurons also upregulated in the roof plate pretty much as they are in the bioinformatic analysis, and so are two markers of neural crest cells. So what we have basically in our roof plate is crest, roof plate, and DI1 identities. Now, the question is, are they all in the same cells? And to this, we went back to the UMaps, okay, and what I want you to look at is only at those images that are here in the red brackets. So under control condition, there is R sponding in the roof plate, but no snail, and this is the overlay, okay? But under treated conditions, we also have both. So the red dots represent the overlay of cells co-expressing crest and roof plate. The same goes for atonal, which is a DI1 marker, atonal 1, and R sponding, and the same goes for snail and atonal, namely crest and DI1 and roof plate and mural crest. So all the combinations between these three cell types can be seen at the level of single cells. And we did the very same comparison on a quantitative level by measuring the proportion of double positive cells, and we see here in different colors the combination of the three cell types, and in all cases the black boxes represent the treated cases, and only this is the control ARSPO1, ARSPO3, just for comparison. So basically what we can say from all our in situ functional and bioinformatic analysis is that retinoic acid is necessary for the segregation between roof plate and PNS, and between roof plate and DI1 interneurons. And I'll skip this. Separation between fates, both at temporal as well as at spatial levels. How am I in terms of time? Should I finish already? Do I still have 5 minutes?
Yes.
Okay. So, specifically, retinoic acid is responsible for crest roof plate segregation, which is a temporal event via BMP. And the question now, the last one I will address, is how does retinoic acid exert the separation between being a roof plate and being a DI1 interneuron, which is localized immediately ventral to the roof plate. Okay, and this is what we saw before. This is a normally separated DI1 from the roof plate, and this is the lack of segregation. And we know that cells co-express roof plate markers, DI1, and crest markers in this, under these treated conditions. And here comes the slide I meant to tell you before. This is mouse work together with Quentin Schwartz in Adelaide. And what we did here, so we begin the parenthesis with a new signaling system for a second just. We found in this work that the formation of the roof plate and of DI1 interneurons depends on notch signaling. Notch signaling is another very important pathway in development and in cancer, which depends on cell-cell interactions. Contrary to retinoic, BMP, Wnt, which are secreted signaling molecules, notch is a receptor, and its ligands, delta, jagged, serrate, are all expressed in a transmembrane fashion, and notch in cell 1 interacts with delta in cell 2 via cell-cell interactions. So we found, using a mind bomb mutant performed only in the crest cells, okay, that in the absence of notch signaling, mind bomb is necessary for degradation of ligands of notch. There is no roof plate. So we applied all the markers that Chaya found in the transcriptome to the mouse. All of them work. So we see in the wild type ARSPO1, we see Bambi, we see GDF7. The roof plate in mice is ugly, is extremely small. Mamzer אבל קטן. You see RALDH2, you see SLIT1, you see MSX1. None of them is present in the roof plate of the mind bomb mutants. Namely, notch is necessary for the formation of the roof plate. And what do we also see? That we lose the expression of DI1 interneurons. Okay, this is MAT1 or atonal 1. So no roof plate and no DI1. What do we have in the mouse? We have DI2 and DI3 interneurons taking over the dorsal neural tube. Okay, so the cells, it's not that they die, they are taken over by more ventral cell types. So point number one, not necessary for the roof plate. Let's go back to our story. When we eliminate retinoic acid signaling, what do we see? We see that the pathway that is upregulated, that is enriched, really is notch. Okay, and we see it here, you can see here, all the notch ligands, jagged, lunatic fringe, delta, and so on, are enriched in our transcriptome. Let us see how it sees itself in the in situ. This is delta. Delta is a notch ligand. You see it from the boundary between roof plate and neural tube, pretty much like lunatic fringe. None of them is in the roof plate, okay? But they mark the boundary and go more ventrally, okay? Because they are important for neural tube development. What happens if we eliminate retinoic acid? You see delta all over the place. You see lunatic fringe all over the place. You see this hasn't been electroporated, no reaction. This has been electroporated. You see lunatic fringe extending into the roof plate. So this means that lack of retinoic acid causes the expansion of notch signaling ligands into the roof plate. Does it affect activity of notch? Yes, because if we come with a PTP venus, this is a reporter of notch activity. Okay, never mind what it is exactly. We see that it marks the boundary under control conditions. It doesn't extend into the roof plate. So this is roof plate, this is DI1, and the boundary is notch. But look at the dominant negative, there is notch activity in the roof plate, and this is basically quantified here. This is the dominant negative, and this is the control with high notch activity in the boundary. So how do we make sense of it? So bear with me, just this is the last, before last slide. So we know this figure already. We know that inhibiting retinoic acid will cause the expansion of DI1 interneurons into the roof plate. Now, from Chaya's work, we also know that if we overexpress notch signaling, now this is an overexpression of a constitutively active notch, we see the same. We see BAR-HL positive neurons in the roof plate, meaning that lack of retinoic acid activates notch in the roof plate. And this is why we saw lunatic fringe and delta in the roof plate. So this is what Dina did, all the smileys. I don't do smileys, only she. So the idea is the following. Under normal conditions, we proposed, based on all the data I've shown to you, that retinoic acid restricts notch activity to the boundary, thereby creating a separation, a physical separation between roof plate and DI1. If you eliminate retinoic acid, okay, notch activity goes into the roof plate. We saw it at the level of activity and at the level of gene expression, okay? And then we see also DI1 cells in the roof plate. All of this I have shown to you as evidence. So how can we prove or ask whether retinoic acid and notch pathways interact with each other? So basically what you do is a rescue experiment. So what you do is you inhibit retinoic acid, but you also inhibit notch activity, and then you are expected to at least rescue partially the boundary between DI1 and the roof plate. If you are with me, this is the last slide. So what we see here, normal conditions, BAR-HL, okay, is only ventral to the roof plate. In the absence of retinoic acid, many neurons are in the roof plate. In the absence of notch signaling that we do by activating a molecule, an important molecule of the pathway called suppressor of hairless, we see that everything looks normal. And when we apply both suppressor of hairless and dominant negative, we can rescue quite a normal phenotype. This is a very heroic experiment because it involves 3 different electroporations that not necessarily will affect the same cells. So we are so happy with the result. So what we see here is that the distribution, this is dorsoventral extent, this is the normal distribution of the control and of the suppressor of hairless. This is what we see as an extension of barrel-gel positive cells into the roof plate, and this is our rescue. So we get a very nice and significant rescue of the formation of the boundary. So conclusion, retinoic acid is also instrumental in setting the boundary between roof plate and DI1 interneurons, this time not via BMP, but via restriction of notch activity. Pay attention that most of the interactions are of a negative type, of a repressive and not of a stimulatory type at this time. So the take-home messages are that crest from roof plate segregate by retinoic acid via BMP, and also wind. This is a whole entire story I didn't talk about at all, but wind is downstream of BMP and it is affected in the very, very same manner. The segregation between roof plate and DI1 and the formation of the boundary between them are regulated via notch restriction by retinoic acid as well. And this brings me to the conclusion that retinoic acid, which begins to be produced in a very, very particular time window in the roof plate, is an essential component of what we call the roof plate organizer. I told you the roof plate is a scaffold. It is a very informative and formative area of the dorsal spinal cord and the dorsal portion of the brain that promotes the segregation of 3 different neural fates, both in time as well and in space. So my last slide is obviously, first of all, to thank Shai Ofek. He was responsible for the notch data in the mouse and for a beautiful transcriptome I didn't show at all. Sari Kagan, one of my lab managers who works in close contact with all of them, and in particular Dina Reckler, who is about to finish her PhD, and she's responsible for most of the work I have presented. Questions if you have, I'll be happy to answer.
BMP is known even before as a precursor of bone and blood-related cells. So could you put that in time as compared to the roof plate formation?
Well, BMP exists in the embryo as early as the gastrulation stage, which is extremely, extremely early. And this is the whole idea, that the activity, let me put it this way, and I'll generalize it not only to BMP. Wind, BMP, sonic hedgehog, FGF, and retinoic acid are the 5 main morphogens that make our bodies what they are. So how is it possible that with 5 morphogens, maybe BMP if you wish, we get all that we get? Everything is precise in time and in space. First of all, regulation number 1, the receptors. Each of them have very, very different receptors in different target cells. Okay? Number 1. Number 2, inhibitors. As we said, as we saw here, there are plenty of inhibitors in each one of the different systems, which are cell type specific and temporally specific. Third, you name it, combinatorics between them. Okay, how BMP inhibits notch or how BMP stimulates notch and the interactions between them. So you cannot put it in context. All of them work all the time, but in different contexts, in different manners. Okay, so you have BMP everywhere. BMP, you have it in the bone, BMP, you have it in the sclerotome, BMP, you have it in the lateral plate mesoderm, BMP, you have it in the dorsal. But in the central nervous system, It is highly specific to the roof and to the dorsal portion of the system, not in motor neurons, not in interneurons. So in every place, they keep a very, very specific type of localization. Okay? It is antagonistic to sonic, for those of you who Baruch is here. Okay? They do dorsal-ventral patterning in the tube and so on.
You mentioned that cell migration is equivalent to malignancy
Metastasis.
metastasis in tumor cells. I mean, the evidence for this?
Oh yes, plenty. It's a very fashionable field of research. We know, for example, when we found a network that underlies the EMT of neural crest cells, it turned out, this was found in 2000 up to 2004. Our first paper was 1997 or 8, I remember. And then it was found that BMP, for example, and Wnt signaling in particular, which is a proto-oncogene, are important in metastasis. Okay, so not only that, BMP, what does it? BMP, snail is one of the crest markers. Snail inhibits E-cadherin. E-cadherin keeps the cells together. When you have metastasis, you have downregulation of E-cadherin. BMP downregulates E-cadherin. Okay, so this is a kind of common language both for developmental systems, not only crest, in the heart. You have endocardial cushions that will cause the separation of the heart into a 4-chambered structure. BMP is responsible for the lamination of the mesenchymal cells that generate endocardial cushions. Okay, and you name it, more and more and more. So yes, there is a very, very strong equivalence, not only in terms of the upstream pathways like BMP, Wnt, and so on, but also in terms of the downstream effectors of the EMT process.
And the morphogenes are similar also?
What do you mean by morphogenes are similar? BMP is a morphogen.
Retinoic acid.
No, retinoic acid acts more like a differentiation factor. No, there are specific morphogens like BMP and Wnt, which are EMT factors. Each one with a different mechanism, okay? Retinoic acid is not an EMT factor, on the contrary, it would cause re-epithelialization of cells. Not necessarily, it depends on the context. Okay, sometimes it decides between different fates, sometimes it can be an EMT factor, sometimes it's a proliferating factor. Everything is contextual here. So 5 molecules making an embryo, come on. Yeah.
Chaya, you began with the concept of states, that we move from state to state.
Yes.
And then you gave us maybe a dozen or 2 dozen molecules with all with cute names, Bambi, snail, socks, and all these
All
things.
come from Drosophila. Nothing is mammalian specific.
Well, I don't think retinoic acid works the same way.
No, retinoic acid.
acid.
And I suspect that this dozen could multiply into thousands, maybe many thousands, if you made any little bit of effort. So the whole thing strikes me less as moving from state to state than something like the winds on the planet, which are so complicated that supercomputers can't even get close to predicting them. Are we ever going to actually understand how the system really works?
Absolutely. You can ask Nomi. We believe in that. Yes, absolutely. Because basically using single cell approaches, okay, you can you have to do 2 things. Using single cell approaches, you see the trees, you don't see the forest. And using in vivo approaches of perturbation, you'll see the forest and you won't see the trees. So it is only by combining these 2 types of experimental approaches that we are going to be able to establish a network of genes, okay, and we can go in a network between the 10 genes that I could show in our network to, I don't know, hundreds of genes, but until you don't validate the fact that they are indeed involved in the process of question, it says nothing. It is good to publish papers today and tomorrow, but not the day after tomorrow. Yes, I think so. Maybe not all of them, but in development, this is the beautiful things. As I said, things change from state to state to state. So experiments are very difficult because you have to follow them in time windows, which are very, very specific, and not all the systems. The mouse is not very suited for that. In that sense, the chip is suited for that because we can begin an experiment at day 2, finish it at day 2.5, begin it at day 2.5, finish it at day 3, and then really create time windows in which we can freeze a certain state and study how molecule A will affect state B or state A. So yes, it is possible, but it's a hell of a work. Okay.
So maybe related to this last point, for example, you said that RA inhibits BMP, BMP inhibits products inhibit RA.
Right.
So how can you get this temporally precise switch, state change,
Very good
given this?
question. So what we think, you remember this movie in which crest cells leave and roof plate cells come up. So we believe that by leaving, all the time when you have FOX, crest markers, in the pre-migratory stage, in the dorsal neural tube, you cannot have roof plate markers because you have FOX and snail and SOX and DLX, etc. Okay? But the moment these cells begin leaving the neural tube, okay, FOX disappears with them, snail disappears with them. So the neural crest markers disappear, enabling, okay, the roof plate markers to come up. So it is a matter of mechanics together with molecular regulation.
But to leave the neural crest, you need a downregulation of the cell adhesion molecules, for example, right?
Of which molecules?
The cell adhesion molecules that
Absolutely.
right? So but
Absolutely.
then somebody has to do that
Yes.
so that you can leave. So there is some sort of a breakage of this deadlock.
Oh yes, absolutely. BMP via Wnt signaling, what it does, it inhibits N-cadherin, which is the cell adhesion molecule that couples all the cells together in the pre-migratory cells, and it does it via what we call beta-catenin signaling. Beta-catenin is bound in the membrane to N-cadherin. When we activate BMP and we activate downstream Wnt signaling, beta-catenin is activated, it leaves the membrane, it goes to the nucleus, and by leaving the membrane, N-cadherin is dispersed. And then the cells can come out. It's not only N-cadherin, Rho GTPase, there's all kinds of molecules. Absolutely true. Yeah, we have paper. 10 years ago. No.
Last question.
Just a small comment first, that it's really fascinating to see all these genes because we see actually in the aging brain in response to Alzheimer's pathology the same factors coming out, which initially kind of surprised us. We keep going to the developmental work and it seems like they're reprogramming, especially glia cells, reprogramming the response, potentially going to maybe to these more embryonic states, kind of changing these cells.
Yeah, because then you need diapers again.
Yeah, exactly. And but I was wondering, so all of these genes, they're kind of inhibiting the previous program and starting and kind of transitioning to a new program. I wonder if you also know, so who are the factors that are maintaining the cell state of, because generally they reprogram to some state where they stay in that, especially, thinking of human, these cells, they would stay in the same state for years.
Well, the point I'm trying to make is that here nothing stays in the same state. Okay, and I haven't spoken about roof plate. The roof plate appears in the avian embryo on day 4 of incubation. Okay, the radial glia appears on day 6, 5.5. So it's 1.5 days, it does its job, and then it goes on and on and on and on. Nothing stays in development. Everything is a kind of intermediate state. It's development is like preparing yourself to maturity. You don't have much of an equilibrium state in development. You have a lot of entropy.
So all of these factors, if you like continue the timeline and reach maturation,
Yeah.
all of these factors are, if the development goes correctly, all of these transient factors go away, or some of them stay to maintain the
Some of them stay, some of them go away. We are beginning to understand that with the work of Susie, another new PhD student in the lab. We studying radial glia, meninges, ependyma, interactions between them, and we see some of the genes going down like that, and some others remaining very, very strongly. But we don't understand what they do there. I assume that they do function 1 in the roof plate, function 2 in the ependyma, okay? Except for wind, which does all the time the same thing. It causes proliferation.
Okay.
תודה רבה.
Thank you very much. אפשר לראות את הדברים האלה הולכים ומתקדמים.
וואו,
שתי
זה
שאלות.
הולך כל כך יפה
לא,
עכשיו. זה לא הזמן שבין...
את יודעת, בטרנסקריפטומיקס, זה שיש לך שני צירים, ביומאפ בדרך כלל קשה לתת משמעות לצירים. זה נורא יפה שאצלכם ציר אחד הוא מרחבי וציר אחד הוא זמני.
אנחנו מאוד מאוד אוהבים את זה.
זה ממש לא מובן מאליו שבאמת זה קורה.
זה נכון.
תגידי, כשדיברת על סטייט, קצת קשור לשאלות של גם של מרשל וגם של אירן בוטר. באופן עקרוני, את היית יכולה לקבל מצב של קפיצות כאלה, נגיד, לעומת מצב של השתנות רציפה, כן? תחשבי על אחד קונספטואלית, כן? תחשבי על יכול להיות נגיד מצב שבו BMP, הריכוז שלו הולך ויורד לאורך הימים שאת במצב של הניורל קרסט, וכשהוא יורד מתחת לאיזשהו ריכוז מסוים, אז הרטינואיק אסיד מנצח, וזה הופך להיות
which is not the case here.
או שה-BMP במצב קבוע וקופץ לאפס.
אנחנו יודעים את זה.
כן, אז זה...
הוא לא קופץ לאפס, הפעילות שלו קופצת לאפס, הוא ממשיך להיות מסונטז ברמה מאוד גבוהה, כי הוא לא עובד יותר על הרוף פלייט, הוא עובד על התאים שמתחת. זה המצב, זה הפקט.
לא, אבל מבחינה גנטית, השאלה מבחינת התבטאות הגנים.
התבטאות הגנים ממשיכה?
האם את רואה, כי כשאת מדברת על סטייטס, את היית רוצה לראות מצב שבו המפות האלה של הסינגל סל טרנסקריפטומיקס, כן, הן קבועות במשך 24 שעות, ואז הן קופצות לאיזושהי תבנית אחרת.
אני מעריכה שאנחנו נראה.
ואז את תראי את התבנית האחרת במשך איזשהו זמן, ואז זה יקפוץ לתבנית אחרת.
בהחלט.
בשביל זה צריך ניסוי שבו את בודקת את הדברים האלה כל 4 שעות, לא כל...
אבל אל תשכח, קודם כל זה נכון, ולך תעשה את זה, אתה תמות. אבל אל תשכח, לא, גם טכנית זה קשור.
אני לא אעשה את זה, ואני אמות בכל מקרה.
בכל מקרה, ולא נעשה את זה, גם אני לא אעשה את זה, כי זה בלתי אפשרי, יש וריאציה בין חיות. אבל מה שקורה הוא שצריך להיות פה, לכן זה מה שהיה לי כביקורת על נעמי. טרנסקריפטומיקה, וגם אמרתי את זה בסוף, טרנסקריפטומיקה זה הוא אומר לך, יש פה גן ברמה של RNA. האם הגן הזה הוא פעיל? האם הגן... יש המון שאלות הלאה.
לא, אבל...
אתה לא יכול רק מהדבר הזה להשיג שום דבר.
לא, אני לא, כן, אני חושב שזה יותר טוב מה שאת אומרת. ברור שצריך ולידציה, אבל זה נותן לך חתיכת מידע.
בוודאי, אחרת לא היינו עושים את זה. תראה, מה שאנחנו יודעים זה שכל מה שאתה מתאר הוא נכון. יש לך